Hi, everyone. This is the demonstration for Lab 5, the Chemistry of Life, Experiment 1, um, which is on proteins. That it's called testing for proteins. Um, as I did for Lab 2, the last lab in Chapter 2, I'm going to go through and make sure we have our materials, and then I'm going to go through the procedure. I'm just going to stop before I do the very last part so you can't see what the results are supposed to look like. Um, let's see. Um, in Lab 5, when you click on the lab, to the right, there's what's called a lab workbook, and that's what you're going to fill in as you do this lab. Um, you also are going to need to have boiling water. So I have started my water boiling over here on this hot plate. Not boiling yet, but maybe by the time I need it. But you're going to need boiling water, and you're going to need an egg. You'll need to provide your own egg. So for my materials, I need 5 milliliters of Biuret solution, and I've got that. That's a blue solution. Biuret solution is an indicator. I don't know if you can see this. I tried to write it as dark as I could, but it's the word indicator. Um, Biuret is an indicator, and an indicator is a chemical, and it will change color in the presence of another chemical. So biuret is an indicator for protein. Biuret is naturally blue, and it will turn some shade of purple in the presence of protein. It may be more pink, it may be lavender, it may be a violet color, maybe deep purple, but it's going to be some shade of pink or purple if there is protein in the solution. So you'll need the Biuret solution. There are two little dropper bottles of this solution. You just need one. And then you will need glucose solution. You will need unknown solution. It's labeled unknown solution. You will need a packet of gelatin. It's a brown packet inside of a clear packet. And you will need the permanent markers so you can label your test tubes. You will need two 250 milliliter beakers. You will need a 100 milliliter and a 10 milliliter graduated cylinder. You will need 5 test tubes labeled 1, 2, 3, 4, 5. And then you will need 5 pipettes. And you need to make sure that you do not use one pipette in one solution and then use that same pipette in another solution. You don't want to take a chance of contaminating the solution. So stick with using one pipette per solution and don't reuse the pipette once you finish with it. A good rule of thumb is just to throw it away. They are disposable. I think I got it. Okay. And the test tube rack. So now when we go to the procedure, label the 5 glass test tubes, 1 through 5. That's step 1. And step 2 is to create an albumin solution. Now, this is, let me move this stuff out of the way, because this is really more of a cooking technique. Albumin is a protein that's found in human human blood. It is found in egg white. So that is where we are going to obtain our albumin is from egg white. I don't know if this shows up, but this little box is here. So some of you may not know exactly how to separate it. So I'm going to show you. You're going to crack it a little bit on the edge and then hold it over a container. Notice I got 2 containers in front of me. You're going to hold it over a container and hold the bottom part, keep the yolk inside the egg, and then just kind of go back and forth with it until the egg white is out. And then you're just left with the shell and the yolk, and you can throw that away. Or you can use it in cooking if you like. wash them better after this. So now I have my egg white, and it says to prepare the albumin solution by mixing 1 egg white with 25 milliliters of room temperature water. Now, I already had my water out that I got out of the faucet, and I had it sitting out for a little bit so that it could get to room temperature. So now I'm going to put this in a 250 milliliter beaker. So I don't really need to pipe it. I can just take the albumin, put it all in here, and then I'm going to measure 25 milliliters. You can do that with your 10 milliliter. You can do 2 10s and 1 5 if you would like. Or I'm going to keep it simple and use my 100 milliliter graduated cylinder to measure 25 milliliters of room temperature water. A little bit more. Remember to get on eye level. And now I went over. Still a little bit over. All right, close enough. So here's my 25 milliliters of room temperature water. I've added that. And then to make my solution, it just says to mix, so you could stir. You could pipe that in and out. But the albumin is kind of thick, so it's hard to do that. But you can just stir it. And then it says to add 5 milliliters of this to test tube 1. So you're going to squeeze the bulb all the way and then just draw up. 5 milliliters is a full pipette. I couldn't get exactly 5. You can do this and have more time than I do. So just make sure you want to go all the way up to the top mark on the pipette. That's 5 milliliters. And if you can't get it all at one time, just like get 3 milliliters and then 2 milliliters. Just make sure your total is 5. So that's test tube 1, has the albumin solution in it. I've already told you it's a protein, so we expect it. I've given you a big hint there on what to expect. Step 3 is to prepare the gelatin, and you will do this in your second 250 milliliter beaker. And this is why you need the boiling water. Now, this step is extremely important. This is the 3rd time I have made this video, actually, because the first time I did it, I forgot about the fact that gelatin forms really fast. This is Jello. So without the good tasting stuff, it's just the gelatin part. So I kind of forgot that it forms very fast and you can't mix anything in a solid gelatin form. So you need your water to be very hot. You need it to be boiling. And you're going to empty the entire packet into the beaker. Then you can just throw it away. And then, actually, we're going to pretend that my water is boiling because of time. But make sure your water is boiling. What you might want to do is put it into like a tea pitcher or something that has out on the end so it's easy to pour. Because you're going to measure 50 milliliters. You've got to be able to pour it into your graduated cylinder. Now, you can use your funnel, but the problem is you might have to use the funnel for another lab. So just be careful. This is like a lab safety issue. Just be very careful with the hot water so that you don't get burned. I'm going to measure my 50 milliliters of what's supposed to be boiling water, but obviously isn't. But it does need to be boiling hot water because otherwise your gelatin will form and you won't be able to get any results that So you're going to do 5 milliliters of that gelatin solution into test tube 2. And the rest of this you want to proceed with as quickly as possible, like I say, so the jello won't solidify. The next one for test tube 3, you want 5 milliliters of the glucose solution. So I'm going to get 5 milliliters of this into test tube 3. Discard my pipette so I don't reuse it. And then I'm going to get 5 milliliters of just water in test tube 4. You can use the 10 milliliter graduated cylinder if you want to, but you can also use the pipette. I'll do it like they say, because you may need that pipette. Probably you will. They don't tend to give you extra supplies. So I've got my 5 milliliters of room temperature water. This is used as your control because you know that this is a negative control because you know that's going to be a negative test. You know water doesn't have protein. And then the last one is unknown solution. For the unknown solution, the easiest thing, you're going to empty this entire bottle into test tube 5. So you can actually pop off the tip of that bottle and pour all of it into test tube 5. This is my 3rd time doing this video. I've already used my unknown solution. So if you notice that, good for you. But that's how you do it, okay? Then you're going to take your Biuret solution and you're going to add 1 milliliter of Biuret solution. I'm sorry, before you do your Biuret solution, this is very important. Step 7, record the color of every one of these solutions. And the colors are going to be pretty much clear, but some of them are cloudy. So some are completely clear and some are cloudy. So just make sure you record the initial colors in your table, in your lab workbook. And then step 8, make a hypothesis about which colors each test tube will be. It will be blue or purple, basically. You think it has protein in it or not. And then after that, you're going to pipe that 1 milliliter of Biuret solution into each test tube. So you can measure 1 milliliter with your graduated cylinder. All we've had in here is water. Or you can take the top off. You can pop that little top off of your Biuret solution, and you can use your pipette. The 1 milliliter mark is the lowest mark. And I'm going to put this in the water, okay? We all know that's going to be the control. So I don't want to show you what the other ones are going to look like. But you're going to do 1 milliliter into every test tube. And then you are going to mix. And the way that you mix the contents of the test tube, I'll show you with test tube 2, is you're going to hold the tip of it. Now, one other thing, and this will mix the contents without you spilling anything. But one other thing that I want to tell you is you may want to go ahead and do your hypothesis before you start the lab. If you write down your hypothesis of which color you think each test tube will change, you won't have a chance of your jello hardening, because if the jello does harden, you're going to not be able to mix your Biuret with it, so you won't be able to do your test right to start over. And last thing is to make sure you take these test tubes and yourself, when you are finished and you've mixed all the test tubes with the Biuret solution, you're going to take a picture of yourself with the test tube rack and the test tubes and the different colors that they change, because that is documentation for me that you actually did perform this lab. And if you have any further questions, just let me know. There are some pre-lab questions and some post-lab questions in the lab workbook, and you will be finished with this lab.